And Koen, you can take it away. You are muted and we can see your screen. You're still muted.
There we go.
Okay, great.
Yeah, okay, sorry. Thank you, Sharon, for your kind introduction. Today, I will give an update on work that we've done in the last 2 years, in which we optimized an assay to determine plasma pyrophosphate concentrations. And so we developed an assay to really accurately determine inorganic pyrophosphate concentrations in the blood circulation. And what kind of molecule is inorganic pyrophosphate? It's a small molecule that prevents mineralization. And mineralization underlies the clinical manifestations in PXE. And individuals with PXE, they have low concentrations of pyrophosphate in their blood. And that is responsible for the clinical problems that the PXE patients have in their eyes, in their skin, and in their blood vessels. And it's interesting to realize that pyrophosphate, so the molecule that's low in PXE patients, is needed outside of cells. And it's also interesting to note that kilogram quantities of pyrophosphate are produced inside cells, also in individuals with PXE. So it's not a problem of having pyrophosphate, but it's having, PXE is not a problem of having too little pyrophosphate, but having too little pyrophosphate in the extracellular environment, so in the blood. So what you all already know is that PXE is caused by biallelic mutations in the ABCC6 gene. And that means that there's no functional ABCC6 produced. And the function of ABCC6 is to get ATP from the liver into the blood. And in the blood, the ATP is the precursor of the mineralization inhibitor pyrophosphate. So you need to get ATP outside, and then it's converted into pyrophosphate. And then the pyrophosphate can prevent the problems in eyes, blood vessels, and the skin. And as already said, reduced ATP release from the liver is responsible for the low pyrophosphate concentrations in plasma of PXE patients. So I also put that in a schematic overview. So ABCC6, the protein that's missing in people with PXE, is involved in release of ATP. And then the ATP is outside of the cell converted into pyrophosphate, which is needed to prevent the clinical problems seen in individuals with PXE, and another molecule, AMP, but that other molecule is not that important for the current presentation. And the reduction in ATP release in the absence of ABCC6 results in PXE patients in low amounts of pyrophosphate. And you can see that in this graph. So in individuals that are not affected, there's more pyrophosphate in the blood. So now that we know that plasma pyrophosphate concentrations, low plasma pyrophosphate concentrations underlie the problems in PXE patients, it is important to know what's the normal plasma pyrophosphate concentration, because then you can try to raise these plasma pyrophosphate concentrations in PXE, in people with PXE. But the problem is that if different laboratories determine what's normal, they find different values for the plasma pyrophosphate that's supposed to be present in unaffected individuals. And that's caused by the fact that it's very difficult to accurately quantify this molecule, pyrophosphate, in plasma. And over the last 1 or 2 years, we have developed a robust method to try and overcome this problem. And our assay that we have developed is inspired by fireflies. I think these are magic bugs that you can see in summertime that produce light. And they do produce light using an enzyme that's called luciferase, firefly luciferase. And this luciferase makes light with the help of ATP. And we actually made use of this enzyme that's produced by the fireflies to detect pyrophosphate in plasma. So pyrophosphate is formed out of ATP. That's what I explained earlier. But you can also reverse that. You can produce ATP out of pyrophosphate. And that's what we did in this current assay. And then the formed ATP we could detect using the firefly luciferase. And you can detect it because then light is produced and you can measure the light. And that is then a measure for the amount of pyrophosphate that's present in plasma. And this is an example. You don't have to see all the details. It's just to give some kind of idea how the assay works. This is an example of such an assay. So on the y-axis, so this x, you can see the amount of light produced in the assay. And on the x-axis, there's a time scale. And if we add plasma, so a blood sample, to the incubation mixture, then we see that, and this is a blow up of this area, and a zoom of this area, then we can see that the amount of light produced is increasing. And that means that there's ATP present in the plasma sample. And that you can see over here by the increase. And then we use a standard to quantify the amount of ATP. The same we do after conversion of all the pyrophosphate in ATP by this enzyme ATP sulfurylase. So then you convert pyrophosphate back into ATP. And you see an increase in the light produced. And again, we add an ATP standard and we can calculate the amount of ATP and pyrophosphate. And then by just simple subtraction, we get a very accurate value for the amount of pyrophosphate in plasma. So that's what we did. And then we applied it to different plasma samples to see how robust, how good our assay was. We had 3 different plasma samples. And we determined every day for, let's say, 9 days or almost every day, we determined in the same sample the amount of pyrophosphate that was present. And what we found was that the values that we got were very similar every day that we did the assay for all these 3 different plasma samples. So that shows that the assay is really very precise and accurate and also very reproducible. So we hope that this will help to find out what the normal plasma pyrophosphate concentration in individuals is that are not affected by PXE. And that should help set some kind of level to which we need to increase pyrophosphate concentrations in people affected by PXE. Because there are many studies will start to increase these plasma pyrophosphate concentrations in PXE patients. Another, and this is my last slide, another way in which we tested how well our assay performed compared to previous assays is in a set of samples that we collected from an animal model for the disease, from ABCC6 knockout rats. And we found that our assay gave better results than the previous assays. But also something that we found out in these assays is that it's also very critical the way you collect the blood from these animals and probably also from humans, that it's really crucial that this is performed in a very standardized manner. And that's needed to get very accurate values for plasma blood pyrophosphate concentrations. So that's what I wanted to share with you today. And if there are any questions, I would be happy to try and answer them.
Great. Thanks so much, Koen. So questions about that. I'll just comment that yesterday you heard us talk about trying to find a measurement that was objective for PXE, and this is certainly one of them. One of the hard things about using it as a measure for the clinical trials, and you'll hear this later too, is it's the very thing we're trying to change. And so using it as a marker and not knowing if in fact changing this marker makes a difference in the disease is an interesting challenge. And Koen and Lily and others have done some animal models that have helped us to understand that part as well. So questions, how long, okay, here's a question. How long will it take to make a readily available test with PPI?
Do you mean a test that, for instance, can be done at home? Or is it meant a laboratory test?
Something that would
which
be
you should send in a sample?
something that would be, I'll say, readily accessible. So you don't have to like travel somewhere. You can either do it at home or you can get it at most doctors' offices or something like that.
Yeah, that will be difficult. So I know that people are also working on something that you can just pinch your finger and then take a small sample from your blood and then try to use that to quantify pyrophosphate. And that would be something that might be used at home, but I would think that it will still take years before that would be available. As I said, it seems to be very critical the way the blood sample is collected, the value you get for the pyrophosphate. So if it's done in an incorrect way, then you get values that are way off. So my prediction is that this will be a test, if available, that will only come via your physician, who should take the blood in a very standardized manner. Otherwise, you get values that are meaningless.
Thank you. What's the decay time of the blood sample?
If collected after collection, as you see in this slide, so we collected the plasma. So after preparation of plasma, so plasma you get by removing the cells from a blood sample. If you take away the cells, then the decay time of pyrophosphate is very slow, as you can see here, because here we stored the plasma for 9 days at room temperature and still we get the same value. So after preparation of plasma, it's really very stable.
Great. I'm going to actually skip some of the very technical questions that we're getting because it's from some of the professionals and skip to some of the other.
They can contact me via
yeah,
email.
they could have a conversation another time. Question: can you introduce ATP to the blood and just let it be converted?
That's actually a very interesting question, but it's undoable. If you want to infuse ATP, because you should then infuse it into the arteries, because if you would take it as a pill, it's completely degraded and you will not get any pyrophosphate in your blood. So you need to inject, and preferably you need to inject into the blood circulation, so into the veins. And I cannot imagine that being a good treatment for PXE, because you should do it at least once a day. So I feel there are probably better ways of increasing plasma pyrophosphate, on which we are now working, that are not invasive, because this is something that I would not advise to PXE patients.
Great, thanks. And how about the time of day, whether somebody's done exercise, if they've eaten, that sort of thing?
Yes. So we have checked that several years ago, and to our surprise, we didn't find huge variability. This was done on unaffected individuals. So we took samples in the morning, we took samples in the afternoon after a meal, we took samples just before leaving from work, because this was all done at work. So then we didn't see a lot of variability. I have also looked into exercise, influence of exercise on plasma pyrophosphate concentrations, and we found there was. That this was done in horses. So we had horses run for 20 minutes, throttle, And then we took a blood sample and plasma concentrations increased. We didn't publish this yet. So some habits might increase plasma pyrophosphate concentration, but we don't know how exactly this works. There's also indications that maybe just if you are sleeping, that plasma pyrophosphate concentrations go down, but we should do more extensive studies to really confirm that this is the case.
Great. Okay, so there's a couple other questions. They're very technical, Koen, so I'm going to recommend that individuals get in touch with you. I'll just quickly say the technical aspects of this are phenomenal, and we're very grateful to the center and to Koen particularly for perfecting this, because it'll be very useful and is useful in the clinical trials. And as he said, we are also trying to find treatments that are not as invasive or as difficult as, for example, what people who have diabetes need to go through, because unlike diabetes, people with PXE won't be motivated in the same way. There's no big crash that's going to come if you don't take your drug.
Exactly.
Great.
Can I forgot to
Sure.
thank one particular person, and that's Stefan Lundqvist. Oh, sorry, because he was the one that did all the experiments that I presented today. So he really thought this assay out himself for a major part. So he was really very instrumental to develop this test.
Great. Thank you so much.